Hi, I'm Jan Witkowski from Cold Spring Harbour Laboratory, and this is the occasion of the 89th Cold Spring Harbour Symposium on Quantitative Biology, and the topic this year is senescence and ageing. I'm very pleased to have James Kirkland with me from Cedars Sinai Medical Centre, who talked about senolytics. And I confess to you that I really did not know what senolytics were before I heard your talk. So can you briefly describe what they are?
What senolytics are is they're agents or approaches to selectively eliminate senescent cells. So many of the senolytics that are currently at the point of clinical trials are ones that eliminate tissue-damaging senescent cells. In the context of cancer, we're going to have to use new classes of senolytics which have been developed, or alternating senolytics with a new class of drugs that we call senesensitizers, because we have to get rid of every kind of senescent cell because they can be potentially cancerous mutation harboring.
Yeah. So I recall that you showed a slide of the sort of variety of senolytic agents, and it had vaccines, it even had physical means. Actually, we may come back to those physical ones, but do these agents have a common mechanism by the means by which they kill senescent cells?
There are several mechanisms that, there are several categories of senolytics which each have arguably common mechanisms within those categories. So the first senolytics were agents that we found by asking the question, why do those senescent cells that are tissue damaging, the 30 to 70% depending on the context, why do they survive despite the fact they're killing cells around them? And this made us think back to what happens with certain leukemias and lymphomas, where those cells kill cells around them, but they're resistant to dying, the cancer cells. So we used data that we had from senescent cells, looking at a range of proteins and genes expressed in senescent cells. This was early days. This was back in 2013. And we used bioinformatics approaches to ask, why do these cells survive? Why don't they commit suicide because of the factors they're using to kill cells around them? And by finding some of those pathways that are responsible, we were able to select natural products, repurposed drugs, or other kinds of entities that would transiently turn off their defenses so they'd commit suicide, basically. Before that, we did develop, senolytics based on high throughput screens. We didn't take them very far, though. We'll have to go back one of these days and resurrect them. So we began working on trying to develop senolytics back in 2004, but the actual principle of the first generation of mechanism-based senolytics, that idea came to us in 2013.
So back in 2004, you were simply screening libraries of drugs and looking for the response you wanted.
That was after 2004. We actually started off when our group was in Boston before we moved to Mayo and now to Los Angeles to Cedars Sinai. We started off by using early approaches of fusion proteins, because we work with people in Boston who discovered the first fusion proteins. And we asked if we could use antibodies against senescent cells and couple them to a killing construct. Since then, that's been done. We weren't successful doing that. We tried for several years, and we tried the high throughput screening, and then we came upon the mechanism-based approach, and that led to some of the agents which are in clinical trials. Since then, other categories have been developed, as you mentioned. Some of them, senescent cells, again, something like cancer cells, many of them have a metabolic shift. They have to use sugars instead of fats to survive, the way that cancer cells do. So that can be taken advantage of by using agents that deprive senescent cells of the nutrient and related pathways that they need, and some of their defenses against generation of toxic molecules that can occur if they're not able to use sugars.
Right. So you block the metabolic pathways that are based on sugars. And
yes. So there's another category of senolytics based on that. There are other categories now based on targeting some of the defenses that senescent cells use to stop them being cleared by the immune system. So senescent cells don't usually die on their own. They have to be removed by the immune system after they're formed. And so again, something like cancer cells, many of them express things called checkpoint inhibitors and don't eat me and don't find me signals and things like that. And they hide out from the immune system. But if we either use vaccines, which are being developed, or have been developed actually, if we use agents that block interactions of those things that senescent cells use to prevent detection and removal by the immune system, or if we activate certain components of the immune system, we can also remove senescent cells. So there are multiple strategies now.
Do all senescent cells have this feature, or is it some subset of senescent cells have this effect on neighbouring cells?
It's only a subset of senescent cells that are tissue damaging. And senescent cells are highly variable. It's a cell fate like differentiation or cell replication or programmed cell death. So it depends, their characteristics depend on the cell type that became senescent, how senescence was induced, how long they've been senescent, and the microenvironment. So, but generally, with respect to age-related diseases and disorders, including things that happen in children that are related to senescence, because we're always aging from before conception on. If there is acceleration of these processes and resultant diseases or disorders, around 30 to 70% of the senescent cells in the responsible tissue tend to be tissue damaging and are susceptible to senolytics.
Usually senescent cells get removed by the immune system. What's the advantage to that 30% of cells that resist getting removed by the immune system?
I don't know that it's so much an advantage as, senescent cells are an example of what we call antagonistic pleiotropy. So some things that are good, if they persist or they exist after natural selection has stopped working, can become bad. So senescent cells are essential for a number of things. They're defense against cancer. So when there's a cancerous mutation or cells have divided many times the way cancer cells do, they can enter a state of senescence, which means they stop dividing. They don't die, they still harbor cancerous mutations, but they sit there. And then the immune system clears them. So this is a defense against cancer. Elephants and right whales, for example, have a great predilection to their cells becoming senescent because they've got multiple copies of some of the factors that will make a cell become senescent should those cells have cancerous features. So elephants and right whales, which live much longer than humans, are much larger, have had many more cell divisions, rarely if ever get cancers.
Because of this enhanced...
They get senescence occurring quickly, but the other side of it that we don't understand is how those animals are able to get rid of the senescent cells so quickly. Because you've got to be able to form senescent cells to defend against cancer, but then you've got to be able to get rid of them fast. Because if you don't, they'll mutate, they can escape senescence, and then they come back as cancers. Senescence is also important during development. So the mesonephros, the sort of fetal version of a kidney that drains the effectively urine into the placenta instead of through the urinary tract, has to be gotten rid of before birth so the regular kidney can take over, and that occurs because of senescence in the mesonephros. The thymus involutes because of senescence and so forth. So it's important development, important in childbirth. Placenta contains many senescent cells. They become pro-inflammatory and tissue damaging in the last 5 days of pregnancy, and that's what drives the baby through the birth canal. So you need senescence to occur. What you don't want is senescent cells to persist, and senescence spreads from cell to cell both locally and at a distance. And there comes a point where if senescent cell burden exceeds a certain threshold, the rate of formation of new senescent cells can exceed the ability of the immune system to clear them, and you get this takeoff, and you get what we call multimorbidity. So in older people, you can get 5, 6, 7, 8 conditions. I, as a geriatrician, sometimes see patients on 20 different drugs. Because, at least in part, these fundamental aging processes are taking off, and one of these processes is senescence, and if one of them's activated, it tends to be through what we call the unitary theory, that all of these pathways get activated that are related to aging.
We should finish in the next few minutes. But you were, I was very interested in your discussion about clinical trials. You were really, pessimistic's not the right, you were very cautious about people, about the quality of clinical trials, and that people shouldn't, well, I'll come back to, and so you've set up large international collaborations doing clinical trials in this area.
Yeah, it's beginning to be the case, unlike in other situations, that the world is coming together with respect to how to do these trials and to do them in a responsible way that's monitored by the correct regulatory authorities, where patient safety comes number one, and where the trials are done in a rigorous way and reproducibility is looked for, and we want to make sure we do no harm. That's the first principle of the Hippocratic Oath.
And you said that no one should take senolytic agents until the results of trials have come through. You were really quite strong about that. And which leads me to when I did a Google search for senolytics, the first row of things that came up were senolytic agents being sold on eBay.
Yes.
So first of all, are those senolytic agents that you see on eBay, are they really senolytic agents, or are these companies just using the senolytic name to sell these products?
I think there's a mixture when it comes to what these agents, what these things that you buy over the counter contain. I mean, we've analyzed some of them, and they don't actually have the agent in them that they say is in them. So I think we need some degree of regulation of all of this, and that means, good manufacturing practices, production. It means supervision by regulatory authorities. In the case of the United States, the Food and Drug Administration, or in Europe, the EMA. And we need to use these agents once they're clinically indicated and we have the trial results by healthcare professionals, who are doing this in an evidence-based and careful manner. We're trying to do the clinical trials as fast as we can, but to do them carefully. So what we're doing is multiple clinical trials around the world. We're trying to coordinate them, and instead of doing the trials in the usual one after the other kind of fashion, we're trying to do multiple trials at the same time for different indications with different kinds of senolytic agents. Because I think, again, the starting point for some of these agents will be for serious life-threatening conditions for which there are no good current treatments. And then we can ethically do this.
Yeah. Well, I think we should stop because the next session is going to begin soon. James, thank you very much.
Thank you.